While you're doing that, I'll introduce you.
All right.
Thanks everybody for attending our webinar today, our Sci-Cafe. Emily Fuqua is with us. She recently completed her PhD at Florida State University in December. Her background includes finfish and shellfish restoration using traditional and restorative aquaculture techniques for fishery management and ecosystem restoration. She's worked with culturally and economically important species such as cobia, redfish, oysters in South Carolina and Florida. Her research also focuses on conservation and restoration of important coastal species and ecosystems. Today, she's going to talk to us about oyster restoration projects she's been involved in and around the Apalachicola Bay area. And she integrates oyster biology and restorative aquaculture approaches. And these projects are aimed to support the Florida oyster population, which we think of as very important to our existence here in Apalachicola Bay. She also uses an array of techniques from larval culture and drone mapping, and she'll highlight promising additional and alternative approaches to traditional oyster restoration in Florida and the Apalachicola Bay area. And anytime you're ready, you can share your screen, which is there's a screen icon and a little toolbar there. If you can see it.
It's giving me the webcam, but I can't see a screen icon.
If you go down the little panel, there's a microphone, there's a camera, and then there's a screen, and you click on that little TV-like screen, and it should allow you to share.
I can see the microphone and the webcam, but then it says like questions in chat.
Try it in just a second.
There it goes.
For some reason, I make you a presenter, and then I have to make you a panelist, and I used to do it automatically, but.
All right, let me.
You see it yet?
Yes, I'm.
Right. Might take a minute.
Yes.
All right, I'm gonna get off the screen and allow you to take over.
All right, I might have to, I think my computer wants me to log out and log back in to do the permission,
okay.
So I will be right back.
Okay. And for those of you who are new to our Sci-Cafe, we hold this once a month generally, and it's usually the fourth Thursday of the month between 3 and 4. And we try to get speakers who are doing work in the Apalachicola Bay area and the Apalachicola River watershed. We won't have one in February because we're having a symposium, but we'll pick up our schedule again in March. And if you want to see some of the Sci-Cafes we've had, you can go to our website. Just Google Apalachicola Reserve. The website will come up, and if you look under events, there'll be a category for past events. If you click on that, you can see all of the Sci-Cafes we've had for the past few years. And we've got some great topics on there. If you ever want to suggest a topic, we'd love to have your input, so you can email me I'll put my email in the questions box. So I'm going to let you take over, Emily.
Yeah, can you share that permission with me one more time?
Oh, sure. It says you're a panelist. Why it's not doing this? I don't know. Connect, make a presenter. Try that. showing me the screen. So
All right. I think it's
great.
starting to work now.
Yay. Yep, we
Allow.
can see your screen. Looks
All
great.
right. Slideshow. There we go. All right, thank you, Anita, for the introduction. As Anita said, my name is Emily Fuqua. I recently earned my PhD from Florida State University, and the majority of my projects was many different things to do with oyster biology, physiology, and oyster restoration, specifically in Apalachicola Bay and many of the surrounding systems. And so you'll see work today that happened in Apalachicola, as well as Oyster Bay up near Panacea, Alligator Harbor, and so on and so forth. And so just to give everybody sort of an idea of the different topics we'll cover, and some of the projects I've been a part of, we're going to start with some context. So we're going to give a little bit about Florida's history and what it has to do with shellfish. We're going to talk about the state of oysters and oyster populations today. And then we're also going to talk about the main avenues in which people go about oyster restoration. Then we'll talk more about some of the projects I've been involved with that have to do with research into alternative avenues and additional materials. That's where we'll really dive into restorative aquaculture approaches, what they are, what they're used for, and how to integrate oyster biology and physiology into restoration. So just to begin with a little bit of history, shellfish has been a real keystone in Florida's cultures since ancient times. There is evidence that even Florida's natives, the Native Americans, really valued shellfish culturally. You'll find these shellfish middens in known areas where natives resided that were large piles of shells, and you'll actually also find evidence of sort of primitive management techniques of shellfish in which Native Americans actually used to reshell or culch areas that they fished. And so they would actually place shell back into the water in areas where they were taking shell out of it. And so pretty advanced management time during this. And as we sort of progressed forward into Florida's future, shellfish remained culturally important and became really economically important too. They're the basis of a very large fishery. They're the basis of aquaculture. And so shellfish bring in quite a bit of money to the state of Florida's economy. This sort of the sort of placement of shellfish as a center point in culture and economics, not only in Florida but worldwide, has unfortunately led to the degradation and collapses of many oyster populations. So this map is from the Nature Conservancy, and you can see that based on the color of some of these regions where we find oysters, oyster populations and their habitats range from anywhere from good to functionally extinct. And so specifically in Florida, we, during this survey, tended to be in the poor to fair categories of sort of the state of our oyster populations. Now, unfortunately, since this survey occurred, many of our populations, including the oyster population in Apalachicola, have since collapsed. Many of you probably already know about Apalachicola, but just to sort of give you an overview, the fishery started declining pretty rapidly. In 2012, it sort of officially collapsed, leading to the federal government declaring it a federal state of emergency, a federal fisheries disaster, in 2013 and fishery closure in 2019. That fishery has since been reopened after allowing some time for the oyster population to try and recover. So what happens when you lose oysters? Why would we even bother restoring them other than the economic problems that come about when you lose a moneymaker in your area? Oysters are what we call a foundational species in ecology, meaning that they actually build an entire ecosystem around them. So they'll come in to an area and change the physical, chemical, and biological environment to actually suit them and build an entire system. They also provide some other important ecological services. So for example, they eat phytoplankton and particulate matter. By filtering these things out of the water, they actually keep the water very clean, they keep it clear, and they can actually mitigate eutrophic environments. Eutrophic environments meaning areas with too many nutrients. Eutrophic environments tend to lead to harmful algal blooms like red tides and harmful bacterial blooms that can cause things like major fish kills in the area and anoxic zones. And so they actually help mitigate that type of habitat from forming by keeping the water really nice and clean and clear. So they eat some of these things. They may choose not to eat it, but it doesn't really matter. All of that gets pulled out of the water and then deposited into the sediment, or the benthos, as we like to call it. This creates a nutrient-rich sediment around them for other things to come in and use that sediment, use that nutrient in the sediment, and it helps just sequester some of those excess nutrients. They create large, complex biogenic structures. Biogenic simply means made of living things. So when an oyster creates a reef or coral creates a reef, we call them biogenic structures. And when you create a large and complex structure in the marine environment, a whole bunch of other stuff moves in, right? And so you get transient species like large fish and sharks that come in to hunt, but you also get resident species such as stone crabs and blue crabs come in, and they actually will live in and around the oyster reef. And so these oysters really do just create these large habitats. They create their own ecosystems. Finally, the last sort of ecological benefit that has an economic effect that I'd like to highlight is the sediment stabilization and decrease in erosion. So as you create structure, you can actually attenuate waves, meaning that as waves come into the coast, they knock into the reef instead of knocking into the coastline. And this reef helps essentially dissipate the energy. It weakens the wave and it allows sediment to stay where it is. So that erosion decreases greatly when you have these sort of barriers formed by the oyster reef itself. And so when you lose oysters, you lose all of these services. Unfortunately, there aren't many other types of animals that would come in and provide these services, as you might see if you lose like a predatory fish. The oysters really do sort of build this large foundation to provide many services in which are completely lost when you lose these reefs and these animals, which is why restoration of these animals is so important in this day and age. So what does restoration look like? In oyster restoration, the most common cause and one of the leading causes of oyster population decline is lack of suitable hard substrate. So the reef itself and the oyster shell provides this hard substrate where baby oysters will actually come in and cement themselves on. And so the traditional approach to oyster restoration is supplementing hard substrate and supplementing shell in the system. And this is simply to give those babies, those young of the year oysters that are swimming around in the water column, somewhere to come in and hold on to, somewhere where they can cement themselves down and metamorphose and turn into an adult oyster. Unfortunately, due to the national decline in oyster populations, we are actually now in an oyster shell shortage, meaning that there is a really high demand on the resource that has decreased availability and increased the expenses in order to get shell. And so you might say, cool, I mean, it might be hard to get it, but why was this such a problem for Apalachicola specifically? To answer that question, this is a map of oyster habitat in Apalachicola Bay. So the light blue is the water areas. You can see all the land in gray, St. George, St. Vincent, and the main coastline. The darker blue outline is historic habitat of oysters in the bay. And this used to cover, I believe it was about 16 square miles in total. Well, after the oyster population collapsed, FWC did a large survey of available oyster habitat, and they found suitable oyster habitat in everything that is colored red on this map. And so this is 95% less oyster habitat was suitable for oysters now in the bay. Much of their habitat had turned into sort of sand or mud or shell hash, but it was all sort of bottom habitat that was not suitable for to support oysters any longer. And of course, this is just sort of square area. This does not take into account the vertical height that we had also lost at that point. These reefs used to be meters high. And so the total volume of shell that we would have needed, that Florida would have needed to restore Apalachicola was just absolutely astronomical. Along with that, the loss of the structure in the bay actually can change currents and how they come through. Many of you probably know that Apalachicola Bay has very strong currents. And so putting sort of bound together shell on the bottom in order to restore Apalachicola simply results ultimately in a lot of material getting swept away in currents. And so that just increases again the amount of shell that you would actually need in order to go about restoring oyster habitat in Apalachicola Bay. So that led to a couple of questions. One being, well, where else can we get shell? And this question was a question that my advisor, Sandra Brook, and I had. Turns out people grow oysters. So we have oyster aquaculture farms all along the coastline of Florida. And during the process of growing these oyster farmers actually generate a certain amount of aquaculture byproducts, so things that they can't sell. And a lot of it tends to be seasonal mortality, which just ends up being oyster shell. And so we met up with some farmers who were interested in potentially getting into some restoration work. And we got a grant funded by the Nature Conservancy and Pew Charitable Trusts to start integrating and testing how we might be able to use some of this discarded byproduct, this unsellable material from the farmers in actual oyster restoration. So we have a farmer-led oyster restoration program in Oyster Bay. This is just a pilot program, so it's relatively small. We have a few contributing partners that over the last year have been making repeated byproduct deployments from their farms onto an intertidal habitat in Oyster Bay. And so they get paid to take all of their sort of unsellable product and mortality out to these sites. We've tested a couple of different biodegradable materials in order to bag this culching material, and they place it along their sites. After they place the material, we come out, we monitor the deployments, and we generally check to see how restoration is going. And then we've been using drone mapping at low tide to monitor sort of how this bar and how these habitats are changing over time. And this drone mapping is really cool when we pair it with really sort of very detailed GPS data, which includes latitude, longitude, and elevation. We can actually recreate these reefs in a computer model down to a centimeter scale. And so we're actually able to look at how this reef is changing, not only in a very sort of wide view, but also down with accuracy down to a couple of centimeters. So we're piloting this program. We're trying to see if there are alternative ways to get shell back to use for oyster restoration in the local area. But unfortunately, that's still not enough material to really make a dent in what was needed for the bay. And so another question that came up when agencies were talking about oyster restoration in Apalachicola Bay was, are there other materials that we could possibly use? And there are a few different types of materials nationally that different groups use. Granite is one, concrete is another, but the material that was sort of most available and sort of the most promising, and that the community and shareholders really wanted to look into, was actually limestone. And the benefit limestone has over some of these other alternative materials is that limestone and oyster shell are actually made of the same thing. So they're made of the same molecule. They're a calcareous compound. And so the hope is that the limestone will provide similar sort of structure and similar chemical cues that oyster shell would originally. And so another project that I've been a part of, that I've been helping with, has been testing the effect of limestone versus shell for oyster restoration. At one of the very starting projects at the beginning of a grant called the Apalachicola Bay System Initiative, which was funded by Triumph Gulf Coast, my advisor deployed a very large field experiment to compare two different sizes of limestone, a large limestone and a small limestone, and compare it against shell to see how oysters settled on it and how growth progressed across time. We deployed both at Peanut Ridge and Dry Bar to see how these different environments would also affect restoration and attempts moving forward. So I'm going to show you just some of the results that we've been getting. This has been going on. This is a multi-year project. And you'll see the first set of graphs will compare the three treatments: large limestone, shell, and small limestone. This first graph has oyster density across the three treatments, and what we are seeing is that the small limestone supports a significantly higher density of oysters. So in a given area, the small limestone is going to have more oysters. This next graph is to show you kind of the survival of these oysters that set. All materials support similar oyster survival, so once they get there and they set on these materials, they sort of have the same chance to continue living as time progresses. Finally, we have the average shell height across treatments, and what we're seeing is that large limestone actually supports significantly larger oysters than the other two treatments. And small limestone supports significantly smaller limestone. And so we're seeing a really interesting trade-off when it comes to these materials and the size of the material that we're using, where small limestone might support more oysters, but large limestone supports larger oysters. So just an interesting trade-off of using this material. And limestone is the main alternative material that FWC has sort of moved on with. So they have been constructing restoration reefs across the Cap Point and Peanut Ridge area in the bay. This is a schematic from FWC where you can see they started with some small pilot studies in phase 1, looking at high relief, which is sort of taller reefs versus low relief, shorter reefs. And then they moved into phase 2 and phase 3, building slightly larger plots to hopefully help jumpstart and provide more appropriate settling habitat for oysters. The other thing that we sort of learned from this experiment was that site really does matter. And so now I'm going to show you the same 3 metrics just across the 2 different sites, so that dry bar and the Peanut Ridge area. So first, the first graph is oyster density again, where we see Peanut Ridge has a higher oyster density. The next one was, the middle graph was survival, the proportion of live oysters, and we can see that Peanut Ridge has significantly higher live oysters. And then finally, the average shell height, we see that Peanut Ridge also has larger oysters. And so when you're talking about oyster restoration, choosing your site is really gonna matter. It's gonna make a difference because we are seeing that Peanut Ridge supports higher oyster densities, supports higher proportion of surviving oysters, supports larger oysters, and another aspect is they also tend to get higher recruitment, although I don't have a graph in this specific presentation to show you all. And so Peanut Ridge is potentially going to really benefit from just the supplementation of hard material in the bay. And there is a very good chance that that area could rebound really well with just taking that approach in that area. But dry bar, unfortunately, in order to get that area back on its feet, may need additional sort of resources put into it. So what else could we do for the dry bar area? How else could we sort of jumpstart that area into hopefully being a productive oyster ecosystem again? That is where restorative aquaculture could come into play. So restorative aquaculture is a technique used worldwide for a variety of different species, and it can be a widely successful program. This map just shows you how many fish conservation hatcheries there are in North America. So it's very popular, especially for salmonids in the Pacific Northwest and all along the West Coast, as well as some of the lake trout, brown trout species that you will find in freshwater systems all across America. This figure simply shows the number of marine species released across the world. And at the time of this particular paper, the United States was sort of the 3rd in line for most number of species released in its marine environments. And you can see that that covers a few types of different species, including mollusks and crustaceans and such. So oyster restoration specifically, this idea of restorative aquaculture was really pioneered in the Chesapeake Bay area. So many of you may have heard about the Chesapeake Bay. It had a really huge oyster population that collapsed in the 1800s and 1900s, and it's been a really sort of slow going process in order to get the full oyster population of the bay back on its feet. Many resources have been dedicated to restoring Chesapeake Bay in order to get these oyster populations back. However, the Billion Oyster Project is a little bit more of a recent one where they are taking spat on shell, so oysters that have been raised in hatcheries, and placing them in and along the bay. And this program has successfully restored over 1,200 acres in the Chesapeake Bay. It's been going on for a while, so keep that in mind, with more than 5 billion spat on shell being deployed in the Maryland system so far. And again, these areas, these reefs are providing more than just sort of oysters for a fishery. The restored reefs are estimated to remove and sequester a good bit of nitrogen from the water, equivalent to about 20,000 bags of fertilizer. And it's a service that is actually valued at more than $1 million. And so these animals that are being placed to support oyster restoration in the Chesapeake are hopefully bringing back some of those ecological services that I talked about earlier. Now, during my PhD, I just thought it was so weird how you would essentially just baby larvae in the hatchery, give them the most calm and pristine water, the highest quality of food available, keep them in stable conditions, and then essentially just throw them out into an estuary whenever they were done in the hatchery. And estuaries have all sorts of things that hatcheries don't, right? They have predators, they have diseases, they have unpredictable water quality. And in my mind, I just couldn't kick the idea that it was kind of like raising a child on Bluey and then suddenly showing them a scary movie and expecting them to just be fine with it. And this really kind of prompted some of my research into these phenomena known as carryover effects. So carryover effects are simply when an early life environment affects later life responses. And we see this happening a lot in restorative aquaculture. The most famous example of a carryover effect is actually behavioral. And in salmon, when they are raised in a hatchery and then get released into the wild, many of them don't know what a predator is. And so they don't display anti-predator behaviors. They won't swim away. They think they're safe and sound. And this leads to really high mortality of hatchery animals because they just get eaten as soon as they get put out. And so I really wanted to start digging in a little bit more to see how raising an animal in a very stable environment affects their physiology and their performance and their health overall once they're placed into a much more unpredictable area. And so for one of my chapters of my dissertation, I raised larvae across a large gradient of salinities. And I did find a pretty significant effect of growth across the salinities, where we have lower salinities generating larvae that are not growing as fast, so suboptimal treatments, as well as we go up in salinity, we have sort of this mid-range of salinity that allows these larvae to grow really fast, signifying sort of a more optimal environment, a more gentle environment for them. So I took these larvae all the way through settling, and after I settled them, I placed them all into what we call a common garden, which simply means that they all went into essentially the same environment, still in the hatchery at this point. So this first graph, they were still in this environment when those measurements were taken, but for the second graph, they were actually taken out and now they are all in the same environment. And I found a completely different relationship in the growth of these larvae that were raised in one environment and then all taken and put in the same environment. So again, this larval culture salinity is what they were in about 2 weeks ago at this point, and they have not seen that salinity since. So in this case, I saw that the larvae that came out, the larvae that were actually in the suboptimal treatments at the lower end, once they metamorphosed, they grew nearly 2 times faster than the ones that came out of the more optimal environments, which was a really interesting kind of flip on what was happening to when they were still just growing up as larvae. So at this point, when they were large enough, I put them out into 2 estuarine systems. I used Apalachicola Bay Alligator Harbor and Oyster Bay to compare what would happen if I took these guys and put them out into 2 different environments. And so I looked at growth rate as well as what's called condition index. Condition index for oysters is very similar to a BMI, a body mass index for people, whereas for people, it is a ratio of your height and your weight. Your condition index for oysters is a ratio of your tissue to your shell. And so the higher condition indices usually indicate that you have a lot of tissue, a lot of somatic, what we call somatic reserves, which is usually fat. So these animals have a lot of fat storage. They have a lot of energy stored up in their tissues. Whereas a lower condition index could indicate an animal that does not have those reserves. So it can almost indicate starvation in some cases where their metabolism is running too fast. They can't build up any extra reserves. They don't have any energy to spare. And so again, there's going to be 2 different types of lines on here, the Alligator Harbor and Oyster Bay group. I want to remind you that the larval culture salinity is a salinity at this point that they hadn't seen in months versus the actual environment they were in, which was their outplant site. I saw a marginal difference in oyster growth rate, although this was not statistically significant. The only technically statistically significant effect I saw was in the site. So at this point, the only difference in growth rate was that Alligator Harbor was growing a lot faster than Oyster Bay. However, when I looked into the condition index, what I found was a fun little, what we call an interaction. So larval culture salinity, the salinity that they had not seen for up to 4 months at that point, was significantly affecting their condition index, but it was different, and how it was affecting their condition index was different between the 2 sites. So you can see that across Alligator Harbor, that line is very straight, sort of horizontal across the graph. So it wasn't affecting the animals that were put into Alligator Harbor nearly as much as it affected the animals that were put into Oyster Bay. And so I'll talk a little bit more about kind of what this might mean in a minute, but the key thing to take away here is that the early environment was affecting what was going on in the later environment, but it was different between the 2 outplant sites. So for restoration, oysters are usually put into the field pretty shortly after metamorphosis. So after they're set, it's usually a few weeks in the hatchery, and then off to the estuaries they go. And so that's kind of in the stage I showed you that had that really, that very different relationship and growth rate to what they were raised in as larvae. And interestingly, some of these, the ones that grew slower specifically, so the ones that came out of those optimal larval treatments, could potentially be more susceptible to predation and competition once they are put into natural environments. So smaller and slower growing oysters tend to be more susceptible to predators like stone crabs and blue crabs, and tend to have a harder time in regard to competition. So if they're not growing, they're not taking up space fast enough, and that allows another organism to come in and take up that space. And then also in this project, I found that early environment and later environments interact to shape oyster physiological condition. So these oysters were being affected differentially based on not only what they grew up in, but also the environment in which they found themselves later. Larger energetic stores could help oysters survive sort of stressful, unpredictable times, which you find normally in an estuary. And so certain animals may have an easier time surviving stressful conditions for longer periods of time. So that's great, and that was a lot of theoretical work, but do carryover effects actually have a clear application for use in things like restoration? And that's a great question. I would love to just give you guys an idea of what happened in 2024. For those of you who don't know, Florida underwent a massive, unprecedented heat wave. So there was a record-breaking heat wave in 2024 that affected the entire coast of Florida. Ooh, that's coming in late. And so these purple areas indicate areas along the Florida coast that actually reached up to and over 32 degrees Celsius, which is over about 100 degrees Fahrenheit. This was an incredibly stressful time for Florida's coastal marine wildlife. Specifically, it led to an unprecedented bleaching event in the Florida Keys, and obviously had other implications for shellfish. So one of the main applications of carryover effects is actually a phenomenon that is termed thermal priming. And the idea behind thermal priming is if you expose animals to what is called a sublethal heat shock, so a heat shock that stresses them out but doesn't kill them, that they can actually go into a period where they have a higher thermal tolerance. And so during this period, they can actually handle what would be a lethal heat shock better. And so I really just scratched the surface as the final chapter of my dissertation on thermal priming in oysters. What I did was I took a group of oyster seed and I split them. I had some controls, which sort of just had ambient temperature conditions, and then I put some through a sublethal heat shock. So I stressed them out, but I didn't necessarily want to kill them. After this, I split both groups. I put some in here at the lab at FSU CML, and others I placed into Oyster Bay. I wanted to see if there was another interesting interaction where that later environment might affect how these animals survive during the lethal heat event. I did measure their growth rate, and what I found was that the sublethal heat shock caused animals to grow slower. And this is just an indication that they were stressed. So they were taking this energy that would normally be put towards growth, as you see in the control circles, and instead they redirected that energy towards these thermal effects, and those are the animals that are indicated by the triangles. So I did see an effect of the heat shock in the growth rate, so the speed in which they were growing. And then when I took them and I exposed all of them to the lethal heat shock, I found that the sublethal heat shock significantly increased the chances of survival of the oysters. And so the only oysters that survived were oysters that had undergone the sublethal heat shock prior. None of the animals who were not heat shocked survived the lethal exposure. And so this has applications for specifically marine heat waves and tolerating marine heat waves for oysters that are spawned in hatcheries and may need to be planted out for oyster restoration. Although this is really just scraping the surface, and there's a lot more work to be done in this case. So just to wrap up, I know I went over a lot of different topics. Oysters have been culturally and economically important for a very long time in Florida. And it's really not just oysters, it's also other species of shellfish, such as scallops and clams. Restoration of declining oyster populations requires significant investment, and this is investments of time, resources. It's really a labor of love. But in order to recoup some of those ecological and economic services that they have given us for so long, it's really an investment that it's work worth doing, in my opinion. Incorporating new and alternative approaches could definitely help some of Florida's oyster ecosystems. And moving forward, what I would really love is to use what we know now about Apalachicola, the decline, the collapse, and the restoration moving forward, to be more proactive in protecting some of the other oyster populations and ecosystems and shellfish populations in Florida overall. And so I hope that is what we are all moving towards into the future, and what I hope my work has contributed to for Florida. With that, I would like to thank many people. This was most of my work in my PhD, as well as the ABSI hatchery and field teams, my advisors, my PhD committees, and all of the funding that has come through in order to fund this work. And I appreciate you guys listening, and I'm happy to take any questions that you might have.
Okay, thank you, Emily. Feel free to turn on your camera. And I will, let's see, I see if I can facilitate some of the questions. Folks are thinking. I actually have a question for you. Are any of the experiments still out on Fry Bar and Peanut Ridge?
So those have been wrapped up. I don't believe that material, like other than the material we brought in to monitor, that material stayed out there. It was similar material to what FWC was putting out, and so it's still accumulating oysters to hopefully help some of the areas around the bay.
Yeah, great. And also, let's see. Trying to see if anybody else has any questions. Feel free to come off mute if you have a question. You can just, everybody should be unmuted. Give it just a minute. And tell me again, what was your, you said the smaller lime rock attracted more oysters, but the large lime rock attracted oysters.
They're bigger. So the oysters over time have been reaching larger sizes on the large lime rock.
Great. All right, I see a question here. Let's see if I can get to it. My screen has shrunk down. Okay, this is, can private citizens grow oysters off their docks using spat from ABSI?
So that's a great question. So there, especially like I said, in the Chesapeake, and actually it's a really big movement up in Delaware Bay called oyster gardening programs, where you can actually receive oysters and spat from conservation hatcheries, research hatcheries, for private citizens to grow and to monitor and everything. That's becoming a very large citizen science set of projects. We currently do not have a project like that set up, but there has been talk in the lab about whether there would be interest in a program like that. And other than really there is nothing stopping it other than the fact that we would need to set up a program to do that. But if you're interested, I would encourage you to reach out to the lab, because most of those programs are really sort of spearheaded by the community. And so if the community is interested, there is probably some space for collaboration there, but you would definitely have to reach out to the lab and inquire about the possibility.
Okay, actually, I can help answer that question. We have these called vertical oyster gardens, or VOGs, as we refer to them now. Currently, we have some education programs with school children growing them on some public docks, and we have a list of private individuals who are willing to put them on their docks and monitor them. It's not labor intensive, but we have well over 100 of these to be adopted. So if you would like to adopt one and you live in an oyster, in an area that will be able to recruit oysters, and you can tell that just by looking down at your pilings on your dock, we'll be happy to set you up with hosting one of these vertical oyster gardens. And just a note, if you live anywhere in the bay, it may not be suitable to harvest and eat them. You have to look at the Department of Aquaculture's map of conditionally approved areas, so just to keep it healthy. And I don't see any other questions, that, unless I missed something. Feel free to come off mute. But Emily, I really want to thank you. We appreciate this. And I don't know if you know, but many of you know, but Emily and her husband, Jared, are moving. Where are you moving to again?
Texas. I will be headed over there in February to spearhead some oyster restoration projects going on in the Aransas Bay area and surrounding systems.
Okay, we have a sister research reserve over there, Fort Aransas, so maybe we'll meet up with you again, and always we love to keep in touch with what you're doing. So thank you so much for doing this Sci-Cafe for us, and this is recorded, and you should get a link to it. If not, my email address is in the chat, so feel free to grab that or give me a call. So thank you very much. We really appreciate your time. And good luck to you.
Thank you for having me.